So today I'm going to talk about some of the work I did during my postdoc at the Ocean Frontier Institute and Memorial University, looking at past and future climate-linked loss in the most northerly freshwater fish, Arctic char. And before I get into the sort of nitty-gritty of the project, I always like to thank my co-authors and my funders, for which this work would not have been possible without them. So today we're at the Genomic Forecasting Symposium because we all recognize the importance of using genomic data to better understand how species will respond to climate change. And I really think that the interest in this work is underpinned by a few fundamental principles. And one of those is the idea that in order to preserve and protect biodiversity, we need to predict how species will respond to climate change. As biologists, we understand that biodiversity is so important for ecosystem function and service. Secondly, we know that adaptive genetic variation is so important for dictating climate change response. So there really is a need to integrate an evolutionary framework into these predictions. Now, over the past few years, there's been a wonderful body of work that's demonstrated the utility of genomics for these sorts of predictions in things like bats and birds and plants and all sorts of other taxa, but we haven't really seen a lot of this work on Arctic taxa, and it's an ecosystem that's especially vulnerable to climate change. So we know that the Arctic is experiencing unprecedented rates of warming. It's having devastating impacts on the biodiversity in the region for a multitude of reasons. One being that as species move northward into the subarctic and Arctic, we see that there are changes to predator-prey dynamics and possibly increasing hybridization. We also know that some of the species in the Arctic already have a narrow temperature tolerance. So under a scenario of increasing temperature, it's quite possible that they're vulnerable to loss. And then with sea ice reduction that's widespread across the Arctic, we're seeing new transportation pathways that may actually risk overexploitation of our important resources. And some of these issues are raised by Colella et al. In a review paper that came out a couple months ago in Tree. And they really take this opportunity to advocate for and to call on scientists to come together and put together genomic data sets for better understanding climate change response in Arctic taxa. So we've tried to start to address this knowledge gap by looking at this in an ecologically and economically vital species in Canada, and that's Arctic char. It is the most northerly distributed freshwater fish, and it's often the only fish in its ecosystems at the north end of the range. We also know that it forms the basis of important commercial, recreational, and subsistence fisheries in the region. So char have a whole Arctic distribution, which you can see here in green on the map. But we were really interested in looking at char from a particular portion of their range, and that's here in northern Newfoundland and Labrador, where the environmental gradients are particularly steep. So this is a really interesting part of the planet and especially interesting part of their range. We see in the south, it's much warmer and wetter, it's more of a boreal forest. And then as you move northward into Labrador, it becomes dry and cold and very much like Arctic tundra. So our goal here was really to integrate genomics and modeling to assess both past and future climate change response in this species in this region of northeastern Canada. So to do that, we collected individuals from 28 populations across the range, again in northern Newfoundland and Labrador. And we used electrofishing to do this to collect mostly juveniles. Our genotype data, we had about 16,500 polymorphic SNPs that derived from an 87K SNP array. And then we mined our environmental data from WorldClim. We also derived a few other environmental variables that were particularly relevant to this project, so things like the number of optimal days and the number of degree days, for instance. So the first thing I wanted to do was really try to look at genotype environment associations. So I ran a redundancy analysis, or an RDA, which is essentially just a constrained PCA. When we look at the RDA, we see on that first axis that populations are really ordered by latitude. So we see the northernmost populations on the left and then the southernmost populations on the right. And what I wanted to do with this RDA was actually identify environment-associated loci. And we define environment-associated loci as loci with scores in the 95th percentile on that first RDA axis. And that's largely because that first RDA axis explains nearly 80% of the variation in the model. So from that, we identified a set of 822 environment-associated SNPs. When we look at the position of these SNPs, we can see that they're distributed across the genome. So you can see those SNPs highlighted in red in the plot here. When I look at the genes that some of these top SNPs are located next to, we see they're located next to things like TGF beta receptor 1, which has been involved in circadian clock function in zebrafish, and other genes like PIP-PMB that's been linked to lipid metabolism under increasing temperatures in Antarctic fishes. In our gene ontology analysis, we see similar biological processes that are overrepresented, so things like metabolism and growth, ion transport, and circadian rhythm. So again, genes and processes that we think are involved in climate adaptation, both in fishes and invertebrates more broadly. So the next step was to take this set of environment-associated SNPs and use this to predict how Arctic char would respond to climate change in the future. And to do this, we used genomic vulnerability. Now, this is a metric that was coined by Rachel Bay and her colleagues in this paper in Science in 2018. And it really pulls from some methods and principles from this paper by Fitzpatrick and Keller in Ecology Letters in 2015. But essentially, the term genomic vulnerability is defined as the magnitude of mismatch between the current and future genomic variation. And so here, this metric has been really useful for us. And I've provided a quick little pipeline of how we actually derive this value of genomic vulnerability. I don't have time to go through the pipeline in detail today, but I thought I'd put it up there in case someone wants to stop and look over it. And of course, I'm happy to answer any questions about this. But the idea with genomic vulnerability is that the greater that degree of mismatch, the less likely a population is able to adapt to future climate change. So I'm going to show you some results for vulnerability across our 28 populations. But before I do that, I just want to point out that we derived vulnerability values under each of the 4 different climate scenarios. So first, I'll show you the results from the least severe scenario, which is RCP 2.6. So that means that this is sort of a lower emission scenario. And then it goes all the way up to RCP 8.5, which is a higher emissions or more severe scenario. And today I'm just going to show you the results for the kind of least and the most severe scenarios, but we see the same patterns in those intermediate scenarios that I'm not showing you here. So when you're looking at the map, I've actually plotted mean annual temperature there, but the data points, the larger and more purple or red a data point is, that's a more vulnerable population. So we can see that our most vulnerable populations are in the south and the mid of the range. Now, when we jump forward and look at the results for the most severe scenario, we see that southern populations are exceptionally vulnerable now, and we see that overall vulnerability values are higher under those higher emissions or more severe scenarios. So next, what we wanted to do was look at the relationship between vulnerability and nucleotide diversity. And this is largely because we would expect that populations that are less genetically diverse are more vulnerable. And that is, in fact, what we see. So again, under each of those 4 different climate scenarios, populations that have lower nucleotide diversity have higher genomic vulnerability. So from this work, it's shown us that our southern populations in this study are by far the most vulnerable, and this is especially true under the more extreme climate scenarios. So these results were really interesting to us, and it got us thinking about life history variation in particular. So for those of you who aren't familiar with Arctic char, it's considered the most variable vertebrate. So it has multiple morphs across its range. We were particularly interested in the migratory and the non-migratory morphs. So if you look at this map, the range of the migratory morph is shown in red, and the range of the non-migratory morph is shown in blue, and it extends much further south. Now, I've put a yellow box around where our most vulnerable populations were found, and you'll see that it's actually at the southern range limit for the migratory ecotype. So in our study, we were only looking at this migratory version. We weren't looking at the non-migratory ecotype. But I wanted to kind of take some environmental data and compare current and future environmental conditions. So I ran a PCA, and you don't need to worry about the population codes or the variables here. I'm just going to kind of point out the colored clusters. But what we see is that future environmental conditions for our migratory populations in this study, which are in pink in the middle of the PCA, they're more similar to current environmental conditions at more southern locations where only the non-migratory ecotype occurs. So if you see there are 4 pink data points falling within that yellow cluster, those are our southernmost vulnerable populations for which future environmental variables look very similar to sites where that migratory ecotype has actually been lost. So that suggests to us that we may in fact lose the migratory ecotype at the southern range limit. We may see a range contraction in this ecotype. And the reason why this is a particular concern is because we know these migratory populations are really important as a food source for indigenous communities in this particular part of Canada. So now that we had an idea that, okay, our southern populations are especially vulnerable to future climate change, we wanted to somehow link these vulnerability results to probability of decline and try to figure out whether Arctic char have responded negatively to climate change in the past. So we had a good opportunity to do this because we know in the North Atlantic in the early 1990s, there was this drop in temperature that had a series of sort of downstream impacts. And you can see the drop modeled by that blue line in this figure. And we looked at this paper by Eric Peterson and colleagues, and they looked at different signatures of collapse in the North Atlantic during the same time period. And they found a significant decrease in groundfish biomass, again, during the early 1990s that corresponds to this drop in temperature. Now, this got us thinking a bit because we had access to Arctic char weight data from the same time range. So what we found is the same thing, that Arctic char weight declined during the early 1990s and corresponding to this drop in temperature. So this is the first indication to us that, maybe char had responded negatively to past climate impacts. But to take it an extra step, we really wanted to see if we could detect population decline. So we reconstructed contemporary estimates of effective population size, which you can see here on the plot. And for all 28 of our populations, we detected decline between the second and third time point, which you can see outlined by this black box. And that's between 1990 and 2001. So again, corresponding to that time where we're seeing a decrease in temperature, a decrease in community biomass, and a decrease in Arctic char weight. And when we really look at the magnitude of this decline, we see it's an average about 35%, so quite substantial. So these sort of past climate impact results taken together with our vulnerability results suggest that there certainly is evidence of both past and future climate-linked loss in this species. And this may have some serious implications for life history variation and really for food security in and around that region of Canada. So you may have noticed from the previous slides, but this work is currently under review, but it has got me thinking about next steps and how I can sort of scale up this vulnerability and climate change response work. So I've always been really interested in rocky intertidal ecosystems. I think that's because I'm from Canada and it's too cold to get in the water. So if you're not jazzed about rocky intertidal, you've got a problem. But we also know that these ecosystems are quite susceptible to climate change. We know that increasing temperatures are driving desiccation stress for the organisms that live there. And I think it would be a really great opportunity to take an ecosystem level approach to understand vulnerability and climate change response. I think there are a lot of interesting candidate species in the rocky intertidal. These are a few that come to my mind. These are species for which there are these really interesting interspecies interactions. You might notice that they're also all marine invertebrates. Today I obviously talked about a salmonid, but I've spent most of my career working on marine invertebrates. So if this is something you're interested in and understanding ecosystem level responses and vulnerability, please feel free to get into contact. I'm now at the University of Aberdeen. So I just want to end off there by thanking the symposium organizers for bringing this symposium online and giving me the opportunity to talk to you about my work today. I also want to again thank my co-authors and funders that I mentioned earlier on in the talk. I want to thank everyone for tuning in, and I want to say Happy Canada Day to my fellow Canadians.